So first of all, thank you for coming here. I hope that today's lecture will be very, very interesting for all of us, beginning from students and other persons, including academicians and professors. Let me introduce Professor Leonid Margolisi. He is now the head of section of intercellular interaction in the institute, which is a part of the National Institute of Health in the United States. So we will have two lectures. The first is today, extracellular vesicles and viruses, close relative or enemies. And tomorrow lecture will be for human immunodeficit virus and infection and what can we do to defeat it. And also tomorrow is planned the workshop with master students and PhD students. I have noted that we have already 12 registered students for this workshop. So tomorrow workshop will be in the another room, E207, and tomorrow lecture will be at the same time in the same hall. So thank you again. Thank you, Professor Margolisi. He is visiting us in the part of the Fulbright supported program supported by US Embassy and government. And also we plan next visit. It's also in April or May, maybe. So if you have any interesting proposals for the next visits or plan any activity, you can offer it us. Thank you again.
Thank you very much for inviting me. I'm very grateful to Dr. Zhuravleva and all my colleagues here. I promise that both lectures will be interesting to everybody, and I'm sure that it will be understandable. First of all, because I don't believe that science should be complicated, and good science is always simple. So today I'll talk about extracellular vesicles and viruses. And first I'll introduce the main hero of our lecture. So the most important thing in biology is cell-cell communications. That's why we are sitting here with our bodies, beautiful faces, and not being just a clump of cells. And for this, cells need to communicate to each other to form our body, our organs, our brain. And what kind of cell-cell communication we know? For years we knew something which is called cell contact communication, when two cells come in the contact and send some molecules from one cell to another. It's like we're talking today now in proximity contact with you. The next is communication through soluble molecules. It's like a bottle with some note which you throw into the ocean in hope that somebody who understands the language you wrote this note will pick up this bottle and get some information. So the cells which will accept these molecules will, should I show them? It doesn't seem anyway. The cells which accept these molecules from the environment would use this to change its physiology. But more recently, we are aware, not long ago, about another way of cell-cell communication, which may be most important. That's communication through the package of molecules, extracellular vesicles, which are generated by one cell and accepted by another cell. It's like sending a package to your aunt, your mother, from the post office, which contains many, many things inside, many information, I don't know, a bottle of Tkemali or something like this. And this has exact address on the surface, which only cells to which is that addressed will accept this vesicle and find some information from them. These extracellular vesicles are extremely important for cell-cell communication, and now it's probably the most growing field in the entire biology, entire biomedicine. So this who invented this? Who discovered these extracellular vesicles? Basically, everybody, they're known to everybody. Everybody of you who ever looked in the microscope on the cell culture saw some debris which were floating near the cell cultures. And nobody paid attention for years or decades to this debris, and nobody really funded study of this debris because it's kind of garbage, and no funding agency would fund you to study garbage. But if you look at more closely, so the nature made laugh on us because in nature there is no such thing as garbage, everything is used. And if you look at this garbage with with electron microscopy or scanning electron microscopy on the bottom, you see there are vesicles which are formed by a lipid layer and has something inside. There are two types of these vesicles. Some of them come from inside the cell, from multivesicular bodies at the upper part, and some come from pinched from the plasma membrane, also forming vesicles. There is a confusion in nomenclature. So some of them called exosomes, some called microvesicles, but basically we don't know now what cannot also pathway is very much different. We cannot distinguish. If you give you one vesicle, you give me one vesicle, with all kind of experimental approach, I wouldn't be able to tell for sure where this vesicle came from, inside the cell or pinched from the membrane. So it's called extracellular vesicles. Now exosomes people use the term exosomes to all of these vesicles. I can refer you to our now well-cited review about macrovesicles, which we wrote together with Yoel Sadowski, my colleague and well-known colleague and friend, and we found different challenges which the science have to understand the physiology of this extracellular vesicle. Anyway, challenges everywhere, from how the vesicles are formed, how they're released, how they entrap something from molecules which are important, how they reach other cell, how they recognize the cell, or the cell recognize it, how it goes inside and release these signaling molecules. Also wrote it several years ago. I would say not much more is known, as still the challenges remain. I told you that it's most growing field in my mind in biology. So in 2019 in PubMed, there were 17,000 papers saying extracellular vesicles only in the title, not exosomes, not some, just extracellular vesicles. Yesterday when I look at it, it was 37,620 such papers. And when I woke up in the morning, there were more papers. So I looked in the January 2023, approximately 17 papers were published per day. That's of course huge, and I doubt that anybody of us, me for sure, I do not read 17 papers a day. But as always in our world, formal information is much wider than the knowledge. And still we have the same problems which we formulated, Yoel Sadowski, this several years ago. So with this growing field, and with such an importance of this field, of course one day I am sure that Nobel Prize Committee would come together to decide whom to award Nobel Prize. And as I told you, there is no real inventor or discoverer, with this extracellular vesicle. Most of the Nobel Prize were given for discovery of something. But as I showed you, all of you discovered it when you looked in the microscope. So with my colleagues, we think that Nobel Prize would be given, but of course it's too late, to Charles Darwin, who actually formulated something about, he called gemmules, particles of minute size generated by every cell type in the body containing molecules and serve to communicate them to other cell types. Excellent definition of extracellular vesicles. And maybe the other Nobel Prize laureate should be It's William Shakespeare who in Macbeth wrote, "The earth has bubbles as the water has." And these bubbles are, I'm sure, these extracellular vesicles. So extracellular vesicles are important everywhere. And as this is not full list of extracellular vesicles importance, and we study some of them, in immunity, pregnancy, viral infection, cancer, microbiota, mental health, autism, and even in marine biology, where some algae send extracellular vesicles to another algae, and this is how ecosystem is flourishing. Just to give an example of how important this extracellular vesicle is, even in practical medicine, last year it was published a paper in Cell, the famous journal, based on the fact that extracellular vesicles which generated by particular cells, they carry some protein, some signatures of these cells, and basically you can distinguish sometimes, analyzing extracellular vesicles, which cells generated them. And of course, when something goes wrong, the cell becomes pathological, it's synthesized new proteins, and the idea, which is long ago, maybe you can have diagnosis by taking extracellular vesicle from the blood and find if something wrong with you. That's called liquid biopsy. The idea was long, but actually the best realization was last year, that published in Cell by David Leiden, who took explants of different tumors and analyzed extracellular vesicles and the proteins of extracellular vesicles with some bioinformatics. And then he took 426 human samples from tissue explant, plasma, and other body fluids, and analyzed the difference with bioinformatics, and found that he can distinguish tumor from normal tissue with 90% sensitivity and 94% specificity. I think it's a great accomplishment. And for me, it's a very important paper, even philosophically, because normally great papers are found on great ideas. There is no great ideas in this paper. It's absolutely trivial, but that's the power of current bioinformatics. When you can arrange a lot of data, accumulate them, you can solve many important problems. I'm not very fond of such science because if I wake in the morning, in the night, I cannot think about this without computer, while I prefer some problems which I can think in the night when I wake up about the mechanism, how from A follows B, and that's probably the future of the science. So you should be, young people, you should be aware of this. Actually, the other, I think, great accomplishment of David Leiden, that he can accumulate so many authors for this paper, for different tumors. There are 108 authors with from 58 affiliations. I think it's maybe the record. Extracellular vesicles are important also for regulating cell-cell communication, especially in immunity. So what is the function of them? For example, we know that there is, for immune building of immune response, you need interaction between cells. Like dendritic cells should contact T cells. But with extracellular vesicles, it doesn't need to contact. It sends pieces of membrane, vesicles, to these T cells, and it's with co-stimulatory molecule, it does exactly what dendritic cells do. Also, for example, vesicles which are derived from T cells, which need to, for immune homeostasis or immune suppression, which should reach different cells in the immune system, and now they don't need actually turned out to reach the cells. They can send these extracellular vesicles, which plays the role of the cells, and the cells, NK cells, natural killer cells, on T cells, which received these vesicles, it does not understand whether it's real vesicles or contact with another cell. So that's the advantage. And basically, it's thought now that all T cell, all communications in the immune system are mediated by this extracellular vesicle. And again, like in David Leiden work, when virus infects, for example, T cells, everything goes wrong, and extracellular vesicles become different, and you can analyze them and find what was different. Basically, now we reconsider even other ways of cell-cell communication. So I give you an example of soluble molecules which generated by one cell accepted by the other cell. So these soluble molecules in the immune system are cytokines. It's a broad family of important proteins, of glycoproteins, which regulate immune response. Turned out, and that's our relatively recent work of my lab, that not all soluble cytokines are soluble. We found that some of them are associated with extracellular vesicles. When we separate extracellular vesicles from plasma, we found with multiplex bead assay, basically it's ELISA on the beads, that some cytokines are associated with extracellular vesicles that are not floating as free molecules. And moreover, if you lyse these vesicles, the number of cytokines you record even increase. So some cytokines are inside, some cytokines are attached to extracellular vesicles. And we analyzed, briefly tell you, this system, whether it's important or not, because everything is attached to everything in biology. So we did several hypotheses. We generated and tested them one after another. If you, after my lecture, find another hypothesis, please tell it to me, we'll also test it. For the system to be important, you should first understand how general it is. Important systems are general. So we studied release from tissue, in vivo, in vitro, from cell culture, from explant, and in all of them we found that cytokines can be associated with extracellular vesicles. The system, which is important, should be somehow regulated. So it should be that with one system, one cytokines are associated, with other system is another cytokine associated. And that's exactly what we found. There are free and EV, extracellular vesicle associated cytokines, in yellowish and blue are in free. And look, there are different systems, and one system, like plasma, there is almost all cytokines are free, and another, they are associated with extracellular vesicles almost completely, like in T cells. So it looks like important. And of course, a good system should be somehow regulated even in one system, and that's exactly what we found. There are some monocytes that are stimulated either with poly-IC or lipopolysaccharide, and look how different are distribution of cytokines between extracellular vesicles and free medium. In poly-IC, almost everything is EV associated, while in LPS, almost all of them are free, which shows that the whole term of immune stimulation is wrong. We should not use it, because immune stimulation, not immune stimulation, not immune stimulation. Different stimuli stimulates in different way, and probably physiologically are very much different, the result. And finally, of course, the cytokine should be bioactive, not just floating with extracellular vesicle. And that we found when we took this cytokine carrying EV and put it on the cells which are dependent on particular cytokines. We found that the cells respond by upregulation particular molecules, which shows that this with the cytokines with extracellular vesicles are bioactive. So that work, by the way, solves one of the major questions in the whole field of extracellular vesicles. So how EVs are addressed to particular cells, and what molecules EVs carry to affect the recipient cells. So this, our story about cytokines associated with, in part, I'm sure that only in part, answers at least two of these questions, because cytokines has particular receptors, very precise on the cell surface, and only the cell which has receptor for this cytokine, which extracellular vesicles carry, would bind these extracellular vesicles. And cytokines are well bioactive molecules, So they should affect the cell. So the way how cells communicate with each other. Of course, nature is never based on the one molecule on one cell, because it's otherwise the system is not reliable. I'm sure that there are other molecules for addressing and changing the cells, but at least there are examples which you found of how it works with cytokines. We don't know exactly what the chemistry of this cytokine associated with the surface. Recently in NIH, a colleague of mine found that chemokine act as phosphatidylserine bind also in apoptotic clearance. So it's part of the answer maybe with binding with phosphatidylserine. So these extracellular vesicles, which all signaling molecules, they really form a language. And it's not an analogy. I have many friends who are in linguistics. And sometimes we come together and drink wine or vodka. And what we talk about when we come together, of course, about science. They tell me about their science. I'm telling them about my science. And they were really impressed how the system of extracellular vesicle with cytokines really resembles language. They're mostly structural linguists. One part of the language is redundancy. Redundancy in mathematics means that you can do the same thing with different words, like redundancy of the language. I'm using some particular words now to get a message to you, but I can use in any language other words with the same meaning. So the same is about extracellular vesicles. Moreover, if you're native speaking this language, even by a short part of the word or the sentence, you can guess what's next in this. So you can recognize phrases or words only by fragments in your own language. And that's exactly what we found happens with extracellular vesicles. This is an envelope. IRS is the agency which takes taxes from my salary. So when I get this envelope, I'm sure that something not good is inside. I can already guess. It's not from my aunt asking about my health, not monitoring, but rather something wrong. So that part of the language, you recognize by envelope what's inside. And that's exactly what happens with extracellular vesicles. These are two extracellular vesicles which carry different molecules on the surface, CD63 and CD47. We take them by binding to particular small magnetic nanoparticles. And when we analyzed RNA, microRNA inside, you see how different is this microRNA from the same system but having vesicles with different address. It's exactly like an envelope I showed you. So basically cell can recognize, already looking at the envelope, what's inside, at least with some probability. So all these extracellular vesicles, of course, resemble somehow viruses. And it's really a kind of revolution happens with these extracellular vesicles in virology. Imagine you live in a small town, some analogy, in Georgia in town, or in Italy, or in United States. And that village is, or a small town, is called virology. And you and your family lived for decades, if not centuries, in this place. You know all about ancestors, you know about your great-grandfather, it's his family, and who comes from children, grandchildren, grandchildren from this family. And suddenly you found that you didn't know everything. You suddenly found that your father has another son from another family, from another wife, and all your relatives also live in the same village, in the same town you live. And that more or less happened in virology. We found that there are extracellular vesicles, which we didn't know, which are close relatives of the viruses. And why they are close relatives? The same way as your relatives in your family. because they are generated by the same pathway in cells, and they are generated not only with the same pathway, but also they are physically very similar. They have similar density, and you have similar size, and even similar molecules on the cell surface. So, how to distinguish them? We tried this for many attempts made to distinguish these vesicles from real viruses in case of HIV, human immunodeficiency virus. So we thought that maybe the proteins on the cells and on viruses and the vesicles are different. So unfortunately, most of this, because of the same pathway, they take from the cell very same proteins. So that's pretty old already, several years ago. There are 316 host cell proteins that are associated with the virus when it comes from the cell. And almost all of these proteins are also associated by extracellular vesicles. You don't know what you keep in your mind. However, so we managed to develop a system to analyze, and I'm very proud of this system, to analyze individual antigens of individual extracellular vesicles. You can't do it really by flow cytometry, because flow cytometry is built to analyze large objects. And with extracellular vesicles and virus, basically you cannot analyze with regular flow cytometry. So what we did, we took these magnetic nanoparticles, about 10-15 nanometers, bound them on some antigens, and when they bound to these vesicles, you can stain with another fluorescent antibodies, and in this way you can analyze some antigens on individual vesicles, because they're small. On cells we now analyze about 30 different proteins on the cell surface with advanced flow cytometry. Here you don't have space for this, so you analyze 2-3 antigens. So the same thing, we analyze individual viruses, in this case HIV. We also bind them to magnetic nanoparticles and stain with additional fluorescent molecules. So fortunately it's not us but our colleagues, and as I told you, these all proteins are taken, whether viruses or extracellular vesicles, from the cell surface. Fortunately it's not us but our colleagues understood that there are still differences in some proteins between vesicles and HIV in that case. So CD81 and CD63 are transpinin molecules which span membrane, and they're similar for both viruses and extracellular vesicles. However, there is one molecule which is called CD45, which due by probably some steric reasons, virus doesn't take it. So if you found something with CD45 positive vesicles, we're sure that it's not virus. And look what happens when we do flow cytometry. So in the upper right quadrant, you can see something, all particles which are CD45 positive. So that basically, so in the low one, sorry, that's basically viruses. And then we found something which is CD45 positive in the upper side, upper quadrant. And that's for sure not viruses, means these are vesicles. And then we stained them with the different antibodies, including the protein which calls anti-GP120, which is here, which is most important viral protein. Through this protein, this virus infects cell, fuse with the cell membranes, as I can tell you tomorrow. So vesicles and virus share viral proteins. In that case, the most important one. So now people found that vesicles are taken other proteins from HIV, and the list is on the top, including some RNA. Also HCV virus, human hepatocyte virus C, also has the same feature, like many, probably all viruses. It's also the infected cells generated vesicles which have the important proteins from this. More recently, SARS-CoV-2, the etiological agent of COVID, does exactly the same. it has some viral proteins which are generated by infected cells, not viruses, but viral proteins. So we don't know exactly the function of these extracellular vesicles. It might be that they stimulate some immune response and they are generated earlier than virus comes out of the cell. Maybe they are floating around and alerting our immune system. The immune system is very slow to develop, and these vesicles, which are not infectious, may float around and say to our immune system, look, be sure, soon the virus appear, be alert. Maybe they do something other things, but here, when you look at these extracellular vesicles, then you don't understand what is virus and what is extracellular vesicles. If you ask virologists of the beginning of the 20th century, or look in some dictionary, what is virus? It's everywhere said that virus is a small particles which replicates, goes into the cells and replicates there. Unfortunately, biologists, I came from mathematics long ago, biologists don't appreciate the importance of definitions. So in biology it's very common that people argue about something before defining what they argue about, and one person means one thing, another person means another thing, and there's no argument about. So this old definition of virus is not valid anymore because we use terms defective virus, non-infectious virus. Actually, in the HIV field, 99% of viruses which are generated in an infected body are not infectious at all. They are defective, and infection continues with less than 1% of viral particles which generate in infected body. So what is virus? What is the extracellular vesicles? Would you call extracellular vesicles with viral proteins defective virus, or will you call defective virus as extracellular vesicle with viral proteins? It's not a biological problem, it's a semantic linguistic problem. We can agree on how to term it. But what we thought, and this paper is well now cited, we published it in PNS several years ago, that we thought that there is a gradient, that is not such a thing of virus extracellular vesicles. There's a whole gradient of particles which the cells generate, and on one extreme there are host extracellular vesicles which were generated by cells whether it's infected or not, and on the other extreme there is infectious, truly infectious virus which replicates, and there are everything between them. And that's now with pretty much a common understanding of extracellular vesicles. Some of them carry proteins, signaling lipids, microRNA is important, and there are plenty of examples as microRNA enters the cell and change its physiology. So, as I told you, extracellular vesicles are very young science, unlike virology, which is almost 2 centuries old. But now, I think that virology is a small part of the science of extracellular vesicles, which has many other parts, as I showed you, as oncology, gynecology, and other things. So I advise you to study these extracellular vesicles because it engulfs many, many sciences. So the question, of course, what's viruses, what extracellular vesicles, is semantic. Biological question is whether extracellular vesicles affect viral infection, play any role. By the way, that is a kind, I think, of shame for our science, because the science in virology, which goes for centuries, because generation of virologists use viruses to infect cells and to study the result of infection. Now we know that there is no such thing as a pure viruses, because all of them come together with extracellular vesicles. So the question is how they interact with each other. Imagine that you are a leader of a group and publish something which, with your young investigator, of the action of particular substance on the disease or on the body. And then you come to your young scientist and found that what he put in the system is not a pure substance A, but something contaminated. So what you do, if you are honest scientist, you'll write to the editor and the paper is retracted. I think that part of virology now should be retracted, because we do not know really what was said in this textbook or in the paper is the effect of virus or the effect of extracellular vesicles which affect the system. So to answer this, we need to study how extracellular vesicles interact with viruses. And looks at a simple thing. I'm sure that all here students would suggest experiment how to understand this. So you use one experiment when you have a mixture of extracellular vesicles and viruses, and two controls, one virus alone, another extracellular vesicles alone, and look at the results. And unfortunately, this kind of experiment is impossible to perform, because as I told you, there is very difficult to separate physically viruses from vesicles. They have the same density, the same size, all the same things. So with all kind of separation which we use, is a size exclusion chromatography or stop gradient separation, you probably can isolate extracellular vesicles free of virus. That's possible. But in the fraction of virus, there always will be extracellular vesicles. At least we don't know how to really separate them. So the only thing is indirect experiment, the type I showed you, based on the some proteins which are present in extracellular vesicles and not present in virus. And there are examples when viruses, extracellular vesicles, fight with each other or help each other. I wouldn't go too far in these examples, but give you a couple of them. So one in HIV field, and that's from our work published not long ago. So as I told you, at least a fraction of extracellular vesicles carries this molecule, CD45, not all of them. So when you take with magnetic nanoparticles, with a magnet, the CD45 positive particles, which are vesicles, not virus, and of course other vesicles remain in the system and infect tissue in culture. So you see that amount of viral replication decreased with two different viruses. We checked this. So we think that extracellular vesicles helped virus to infect, and now we take them out, they virus infects not that efficiently. So relatives, of course, do not mean necessarily friends, as you may know in your family. Most of the enemies and arguments goes inside the families, as you know, I hope not in yours. So there are example, I give example when they fight each other, so they help us in terms of infection. And that's again recent our work with lactobacilli. Lactobacilli is part of microbiome of female vagina, which plays a very important role. In healthy women, this lactobacilli, as is shown from many epidemiological work, defend women from infection, vaginal infection, with many pathogens, including HIV. So we really made a similar system with blocks of cultures, block of tissue which we use. We put cervicovaginal tissue in petri dish with a way we know it can be cultured, and infected with HIV with or without lactobacilli. Now we found that in this, as you show in the low curve, that with when you put lactobacilli, there is almost no infection. Unlike in control, we don't put lactobacilli. That was a good work, but now more recently we found that you don't need lactobacilli. It's enough to put extracellular vesicles which lactobacilli generate without lactobacilli, and you have the same effect. With extracellular vesicles, there is almost no, in blue, almost no infection, while control, you have a good infection. So basically, extracellular vesicles, we now think, is part of important microbiome, not only probably in vagina, but in guts and other parts of the tissue of the body. And you know, of course, that microbiome is now one of the most important things people study. It claims that whether you are smart or stupid depends on what bacteria live in your guts. Whether you're obese or not also depends on bacteria, and probably that's also about extracellular vesicles. It turned out that the mechanism of this phenomenon, why extracellular vesicles in vaginal bacteria, lactobacilli, prevent infection, turned out very simple. They just these vesicles bound to the viruses and shield these molecules which are needed for the virus to infect cells. At least antibodies cannot reach these molecules, which proves that they are sterically and probably specifically shielded from the environment. And so these viruses don't have any more molecules to infect the cells. So recently we published a paper in PLOS Pathology about the microbiota as host-to-host communication. So bacterial extracellular vesicles in this case are also a language which bacteria interacts with our body. So as we know, microbiome and all bacteria enter our body after we are born. We're born sterile and then bacteria enter our body, creating this microbiome. So they are like new immigrants. And immigrants in the country, of course, need to learn the language of the country. That's what I did when I immigrated to the United States. That probably some of you do with Georgian when you immigrated to Georgia, if you immigrate. It's interesting that this bacteria do exactly the same. Look, bacteria is approximately 10-fold smaller than cells in our body. Nevertheless, extracellular vesicles they generate are exactly the same size, which shows the importance of this language in communication between bacteria, microbiome, and our body. So as a conclusion, what you should at least remember vaguely from my lecture, that extracellular vesicles, EVs, are produced by all cells and carry signatures of the cells of origins. So if the cells of origin become pathologic, these extracellular vesicles may carry also these pathological molecules. EVs carry cytokines and of course other molecules to other cells, affecting the status of these recipient cells. And this could be lipids, microRNA, proteins, gangliosides, or whatever. EVs are altered in the course of various disease and then can be used as biomarkers, liquid biopsies. That's a very developed field which is rapidly developed now, because you can probably find disease long before you're diagnosed with other methods. At least there's a hope. And EVs produced by virus-infected cells carry viral proteins and affect viral infection. So now more and more particles are discovered by science. So we found that these extracellular vesicles is only part of what we really studied. There are particles which are smaller than extracellular vesicles, like virus. There are particles which are larger, especially generated by apoptotic cells. So there is a whole new world of extracellular vesicles. And what we mostly study, which I told you, is a small segment. So I think we are now in position of Newton, who did a lot of studies of visible light and made great discoveries. But now we know that that visible light is a small fragment of magnetic waves, lengths which go both to the right and to the left of visual light and visual light. It's visual only because we generated in evolution receptors for this light in our eyes, but otherwise other animals can feel other waves. So I think we're now in this stage, but hopefully we know the role of other particles. And I think that's a very exciting world, at least for me. I think that we're now like Leeuwenhoek, who looked in his microscope on a drop of water from the pond and found a lot of animals floating there, bacteria. And we still don't know for most of these microorganisms what's their function, what's their role, for example, if they invade human body. And the same is true about extracellular vesicles. I think it's exciting new world which now opening in front of us, And even if you don't study, even a reading of this in magazine, scientific magazine, is very interesting. Of course, there is another question which is beyond my expertise, is what in this family came first, vesicles or extracellular vesicles, or viruses. Is a vesicle, extracellular vesicle, is a stupid virus which just becomes stupid, or virus is a smart extracellular vesicles which learned how to infect and to replicate. That's similar to what came first, egg or chicken. Then to this, the second question I know the answer. It is in the old Soviet time Russian joke, when the old gentleman in Soviet times, when the deficit of food, with his young small grandson, comes to the supermarket, and there is nothing in the supermarket except something which is not eatable. And the small child said, "Grandfather, do you know what came first, chicken or egg?" And the answer, "My dear, before communists came to power, everything was available." So maybe the same is true with extracellular vesicles and virus. So thank you for your attention. If you have any questions, you can ask now. You can send me an email. You can find my email on the website of NIH. And I promise I'll answer all your questions. Thank you. Before you ask, I must say that I don't know in Georgia, but I gave lectures a couple of years ago in Italy. Students, there are some disciplines, professors sit in the first row, lecturers in the second row, and then students. And only professors and lecturers ask questions. Unlike United States, where everybody jumps out from the back just to ask questions because of curiosity and also because to show your professor how smart you are. In Italy, it's almost different. And I thought, are really Italian students more stupid than American students? I don't think so. And in my last lecture, it was a course, I asked them to send me notes, paper notes with questions, and found a lot of smart questions which people ask, interesting questions. So it turned out it is only a kind of national tradition. I hope this Georgia is more close to American tradition. Everybody who has questions should ask. Elena, please.
I will start, excuse me. So the first question is about extracellular vesicles, which protects of lactobacteria, which protects from HIV infection through the blocking of anti-AGP 120. So it's which part of this vesicle is responsible for this blocking? Only outside parts, surface proteins, or it depends also on the content inside of these vesicles? And the second question will be about the first part of your lecture, connection of extracellular vesicles with autism. Any data? It's very interesting. Thanks.
Yes, your question is absolutely meaningful. So I think the answer is that outside of the vesicles is important, because when we treat vesicle with proteinase K, which shaves all the protein from the surface, it doesn't protect anymore with HIV, against HIV. As far as the second question, I cannot answer. I'm not an expert in autism. I know there are papers. What we studied is a correlation with EVs with depression. So we have published, you see, I am a virologist, cell biologist, and I thought that I end my career quietly as a virologist. But now, since extracellular vesicle spans all biomedicine, I suddenly find myself in the middle of a much wider field, because my colleagues come to me as an expert in extracellular vesicle. Let's do this, let's do this, let's do this. We published recently papers in complicated pregnancies, in different viruses, and even in depression. So we studied depression actually is the difference between two types of patients. Is a man which underwent prostate surgery and then irradiation, or women which underwent surgery of mammary gland and also underwent irradiation. And some of these patients are really depressed because of irradiation, and some are not. And it turned out that the combination, that spectrum of extracellular vesicles are very much different in these two types of patients. We don't know why. We don't know whether it's important for real disease or not. We found only correlation, but still correlation is important, because if it goes before real depression start, you can treat these people in a different way, understanding that about autism, unfortunately I cannot answer, but there are papers. You can search in PubMed.